Welcome to BCRF's Behind the Breakthrough series. I'm Margaret Flowers. Today we're bringing you an update of the AURORA US study with BCRF investigator Dr. Charles Peru. Dr. Peru is a primary investigator of the AURORA US study and a lead author on the recently released article from the journal Nature, where they reported their findings from the first phase of this study. The AURORA US and its partner study, AURORA EU, collectively known as the AURORA study, comprise the largest global effort aimed at understanding the molecular underpinnings of metastatic breast cancer. The AURORA study is the flagship program of the Evelyn H. Lauder Founders Fund for Metastatic Breast Cancer Research, named in honor of our late founder and under the scientific leadership of our founding scientific director, Dr. Larry Norton from Memorial Sloan Kettering Cancer Center in New York. And now I'd like to turn it over to Dr. Peru and ask him to introduce himself to you and tell us a little bit more about the concept behind AURORA and what makes it so groundbreaking. Dr. Peru.
So I'm Chuck Peru. I'm a professor of genetics here at the University of North Carolina, the co-director of our breast cancer program, and also co-director of our computational medicine program, and really honored and privileged to be one of the leaders of the AURORA US project. So what makes this project so special is a number of things. First of all, we're focused on metastatic disease. And as you can see in the paper, it's particularly metastatic disease coming from patients who died of that disease, right? And who've undergone multiple lines of therapy and the therapies failed them. And so, we need to determine what are the molecular causes of that therapy refractory metastatic disease. And so, those of us who have been involved with BCRF efforts over the year, particularly through the Translational Breast Cancer Research Consortium, put our heads and resources together and collected a very unique set of specimens. And one of the unique aspects is not only did we get the metastatic disease, but we also had the primary disease. And so we did a lot of analyses comparing the primary tumors to the metastatic tumors. And when they come from the same patient, that gives us improved statistical power to make new discoveries. And one of the other very unique aspects of this study was that for each of the specimens we collected, and in the end there were more than 50 patients participating and more than 100 metastases that came from all of that, we performed multiple cutting-edge genetic and genomic technologies. So in a typical research study, you might do one of these technologies, like sequence the DNA or look at gene expression changes, but we sequenced the DNA a couple different ways. We looked for gene expression changes using another technology, and with a third technology, we looked at what's called DNA methylation, which is another means of regulating the function of different genes. And many of our most fascinating stories came from really the combination of all three, right? You really needed all three to see, some of the picture that arose, and that was really exciting. And I think there were some important new findings here.
Great, thanks. And you really covered a lot of ground there. I want to step back a little bit in terms of the patients themselves. AURORA US has two phases, the retrospective phase and a prospective phase. And this study really focused on the former, the retrospective. And so before we kind of get into what you found through all these analyses of the tissues, could you explain a little bit to us about, now this was retrospective, so we didn't have new patients enrolled. The team used archived or banked tissue samples from patients that had metastatic breast cancer. Can you, even though those were archived samples and not in new enrolled patients, you had a lot of information about the patients, their treatments, et cetera. So can you talk to us a little bit about what was that patient cohort like? How did it reflect the relevant population, and what did it take to procure these samples that were stored samples?
So, whenever getting important clinical specimens is always requires great effort, right? And particularly for large numbers. And we also knew that amongst the TBCRC institutions that a number of us had been banking these very valuable cases over the years. And again, for many of these patients, we had their primary tumor and not only metastasis, but in some cases, 3, 4, 5 different metastases from the same patient. So we could do comparative studies of, in essence, the same tumor in 5 different locations. And we had some interesting findings from that. This sample set is really reflective of certainly metastatic disease, but as I mentioned, metastatic disease, and also these patients have undergone many lines of treatment. So I think they had an average of 3 lines of therapy or more. And so these really reflect, I think, the patients in greatest need. And in some ways, the patients we know the least about the unique aspects of this disease. So I think we all felt compelled to shine a big research light on this. And here's where I'm very thankful to the BCRF and very thankful to the donors who have supported this effort, because I think this is a critically important patient population to studies. In some ways, I'm almost the most important. And so I'm thrilled that we had both the retrospective phase, which really gives us a running start, right? We didn't have to wait years to get enough samples to do an analysis. They already existed. We just had to organize them and, get the clinical data out of our hospital medical records, which is no small task. But of course, we were able to do that and pulled together a very meaningful set of samples that gave us some important biologic insights. And then again, I think it's really important to know that the study is going on, right? So the prospective phase, we're going to collect even more patients. The prospective phase has some unique aspects that the retrospective phase didn't have. Like, we're doing a few more assays looking at things that are occurring in the blood biomarkers, as well as the tumor biomarkers, and some of the information is being returned to the patients.
Well, that's great. Thanks so much for that background. So let's get into what the group reported in the Nature Cancer article. What were the key findings? Were there any surprises? Did you confirm things that you sort of expected that you would see? What are the big messages here?
So I could, of course, spend an hour discussing the many exciting findings, but I think there were a few that rose to the top. And one of, overwhelming theme was the importance of the immune system and how the immune system, gets diminished in the metastatic setting, particularly in patients with triple negative breast cancer, where, it seemed to be the major thing that changed. Where in primary cancers, there was typically immune system presence and involvement. And then in the metastatic setting, in triple negative cancers, the immune system was much less frequent or, gone almost. And what we saw through our what I call our 3 different genomic assays, again, we needed all 3 to see this story, was that the tumors were evading the immune system in a specific way, where they would turn off a certain gene, which is basically the gene used to sort of expose a tumor to the immune system. So if you remove this one protein, this one gene, suddenly the tumor is cloaked and it can't be seen by the immune system. And 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 so that, that was that was enlightening, and actually suggests for those patients there might be a means to reverse that cloaking. And so those would potentially lead to new clinical trials to prove that that is true. And certainly in the immediate term, we're mimicking that in the laboratory models and and trying to figure out how to reverse that process. Um, in ER positive disease, uh, the story wasn't quite as much about the immune system, but what we did see was that there were a lot of what we would call these DNA methylation changes. Um, and that was one of the 3 genetic assays that we used. And many of these changes were involving genes regulated by the estrogen receptor. And so there was a pretty dramatic shift in some of these tumors, and like what genes were on and what were off, and this was really, pinpointed by using, again, that DNA methylation assay. Maybe one of the third take homes and one of the most immediately practical is, we looked at gene expression changes and gene expression subtypes. That's been a long standing, effort of my laboratory. And what we could see is that in about a third of the patients, the expression subtype changed from the primary tumor to the metastasis. And when it changed like that, it was almost always accompanied by underlying changes in the DNA as well. And so maybe the practical aspect of that is, when a patient presents with metastatic disease and maybe had primary disease years previous, if at all medically possible, we should sample that metastatic disease, because it has at least a 30% chance of being different than the earlier disease. And I always argue we want to study the disease the patient has today and make treatment decisions based on the most cutting edge assays and the sample from today, particularly if it's changing half the time, or a third of the time. For me, that's precision medicine, right? I want to, as best as I can possibly say, this is what the disease is now, and this is what I need to attack.
Right. So there is a lot to think about from these results, and I know that these are early results. There were 50 patients, some with multiple metastases, but given that limitation in terms of size, what can we say about this data when we look at, for instance, what we're seeing from the AURORA EU study? How much confidence can we gain from this, and what can patients take away from these findings today?
So the AURORA EU is a highly complementary study to what we did in the AURORA US. We took an approach of sort of fewer samples with more assays, and they did a larger number of samples with fewer assays, with the goal of sort of using one of those, a primary assay of DNA sequencing to enroll patients in the specific, DNA mutation focused clinical trials. They've also done multiple assays on a subset of their samples. And from what we've seen so far in our unpublished collaborative work, and I think it's okay for me to say this, is, you know, we've validated many of each other's findings, right?
Hmm.
So these are, you know, we have a small sample size, but we can already see some of the same things in the AURORA EU data set. And so we have very good confidence that I will call these are what I call robust and reproducible findings. And so, you know, we should take that and run with it, and now say, okay, what's the clinical implication of this, or and more importantly, what's the clinical utility? What are we going to do with it? You know, many of the findings, unfortunately, are biologically informative but not clinically actionable. And there are some that are potentially clinically actionable, and now we need to show through experiments and then eventually through new clinical trials that the proposed action is is true, and beneficial to those patients. And that's really the next phase that we're entering.
So that would be the prospective study then. Those are some of the things that you'll be pursuing through the prospective study, or are you also in continuing the work from the retrospective samples?
It's Yeah, it's some of both, right?
Uh-huh.
So in the prospective study, actually, you know, for some of the molecular analyses that are planned, we need a larger sample size, and we always knew that, and therefore, some of the answers we are searching for uh, will only come when we've done both the prospective and the retrospective and combine them together. And that's always been the plan. But of course, you stop and take looks along the way, and because the sooner you discover something, the better. And for some of these things that we saw, like these changes in the immune system and the changes in the DNA methylation patterns, we had more than a sufficient sample size to find these. Um, you know, and as part of the paper, we actually also looked to other public data sets to help validate some of the results, and they were validated. And so, you know, it once again reaffirms the value of making these genomic data publicly available so that others can use it and learn from it, and that others can use it to validate some of the findings that they might have in their own studies.
Yeah, on that note, we've got the Aurora US here being conducted through the Translational Breast Cancer Research Consortium, and then Aurora EU through the Breast International Group in Europe. What opportunities will there be for that data to come together? You talk about sharing data and being able to validate data. What are the opportunities for those two data sets to come together and actually converge to to make the findings even more powerful, or new discoveries?
So, you know, BCRF is once again stepping up to the plate and providing a resource to basically coordinate the data from the two sites, house the data from the two sites, and to make it publicly available to the world. Uh, and that should facilitate, you know, more discoveries and faster translation. And you know, I've long said, this is not my data, right? This is research data, and it's for the community to use, because there's so many smart people out there, and there's so many good ideas, you know, not in my head and not in the other people who are immediately working on this. And so we need to get it out there for maximum utility. And again, the BCRF is really, facilitating that. And we've also put this data into the somewhat, I'll say, more standard public repositories. And so, we feel we'll also make an impact by making this data available to the scientific community, and others are going to use it and make discoveries that we didn't make. And the
Right.
data is so rich that it sounds crazy at times to say that, but it's going to happen. I know it will.
I would also add that, just for the sake of our, for our audience, that the paper, Nature Cancer paper, is what we call open access. So that's available without any kind of publication fees or anything like that, or excuse me, subscription fees to access that article. So I mean, that's also really important in terms of making data available for others to validate and to use, etc. As we close, Dr. Peru, on behalf of our supporters who have really championed this effort from the very beginning, and it has been a long process in terms of when this started and getting first data out. And as you said, using the retrospective samples allowed you to kind of get a jump start and start asking some questions and probing some tissues. Um, in the context of the support that we've had for this study, and if you could just really kind of, and you've mentioned it before, that really without BCRF and our continued support, we couldn't move on with this. What what would we risk at this point? You've mentioned several times already just how critical and how important this study has been. What would we risk if we weren't able to continue the what will be more years of research? And when we look at additional patients who are being enrolled in both AURORA US and AURORA EU, what do we stand to lose?
Time in people's lives, right? I mean, the slower the research goes, the more people that will die of the disease, and that's the exact opposite of what we want to happen, right? We want to impact patient care and increase survival, and particularly in this, this critically important area of metastatic disease. We learned a hard lesson, and I think Aurora EU did too, which is, getting you would think it should be easy to get these samples, but it's actually harder to get these samples than it was to get primary cancers. We overcame that obstacle, and now I feel we're really, you know, at the beginning or at the end of the beginning of this study, right? There's so much more to go, and and it's so exciting, particularly with many of the new technologies that are coming on, right? So as we go into the prospective phase, people should realize, again, we're just starting to move fast now, and we need to keep moving fast, and more patients in the study is much better, both for the patients and for the science. And we want to do more cutting edge assays, so there are new technologies we hope to and plan to incorporate, including these what we call spatial imaging technologies. So we can now look at pictures of the tumors and extract information from that. We can look at the location of tumor cells compared to other tumor cells, compared to immune cells, and that gives us additional information, right? So we need to bring to bear more technologies. And of course, you can imagine these things are brand new and more expensive, but I think worth it. And and so again, I just want to, thank the scientific leadership of BCRF, in particular Larry Norton, and again, thank all the donors who've contributed or thinking of contributing to this study. Uh, and to say that, we have an outstanding group of scientists and what we call our molecular working group and our clinical working group, and we're really excited about what we've done and even more excited about what's to come. I do want to end by, of course, thanking the patients for their participation in these studies, because they are making they are the key. And donation of samples today is the key for the next discoveries. And without that, we would be going at a snail pace. So thank you to the patients for all that they've done for us.